Hi everybody. Earlier we had spoken upon the denaturation and the renaturation of DNA, and now let's go into the procedure of COT analysis, the method which has been used to check the amount of renaturation. For this, what you do is you heat a sample of genomic DNA until it denatures into a single stranded form. Then you slowly cool it so that the strands get a chance to pair back together. While the sample is being cooling, the measurements are taken of how much DNA is base paired at each temperature. And the amount of single stranded and the double stranded DNA is measured rapidly by diluting the sample with a buffer, and this slows the reassociation, and then the binding of the DNA to a hydroxyl epitide column is being done. The column is first washed with a low concentration of sodium phosphate buffer, which will elute the single stranded DNA, and then with a higher concentration of phosphate, which elutes the double stranded DNA. And the amount of the DNA in these two solutions is then measured using a spectrophotometer. So I think when you mention like this, it finds to be a little bit complicated. But what you have to do is, when you have a double stranded DNA, that DNA is first broken into small fragments, and then every fragment, like a pool of those fragments, are being denatured by the processing of heating. Once it is heated, what happens after you take it, you immediately cool it to a temperature which is about minus 25 degrees centigrade, less than the Tm value, so that it starts reassociating. And then when it is been taken, what happens, it has been also diluted so that the reassociation has been slowed. Okay, the reassociation or the renaturation of that has been slowed down, and later on this is being loaded onto a hydroxyl epitide column. Okay, and using different concentrations of a buffer, the single stranded and the double stranded DNA are separately eluted from the hydroxyl epitide column, and the amount of the double stranded DNA is being measured by using a spectrophotometer. Now let's see into a pictorial representation of that so that it will be clear. Now here this is in this experiment, a tissue sample has been taken and it is homogenized using a blender, and the nucleus are all pelleted by centrifugation, since it's a plant, and the plastids are all preferentially lysed here. This is a plant which has been shown here. Okay, and the pellet should be free from contaminating organelles, and that has been verified in the case of phase contrast microscopy. Now if you're taking another cell which is not other than a plant cell, you don't have to worry about the plastids and all that, and basically you have to isolate DNA from the organism of interest, and you have to isolate the DNA using phenol chloroform extractions coupled with proteinase and RNase treatments, so that you get a pure DNA sample. After that, the DNA is cut into pieces by using a 450 base pair sonication or a high speed blending, and those DNA fragments are being checked on the gel electrophoresis. Then after that, the sheared DNA is precipitated. You shear the DNA and it is precipitated, and the aliquots of the DNA is dissolved in different levels of sodium phosphate buffer to produce solutions of known concentrations. The DNA solutions are then distributed in a glass microcapillary or a glass ampule, so that each tube contains 100 microgram of DNA. Now the ends of the tubes are also being sealed here. One of the tubes containing known concentration of DNA is selected, and the tube is placed in a boiling water to denature the DNA double duplexes. The tube is placed in water bath at a sample criteria, now for example at temperature minus 25 degrees centigrade, as we told earlier, and the renaturation is allowed to occur until the sample reaches a specific COT value. A specific COT value, as you remember, COT value is a product of the sample's nucleotide concentration into its reassociation time in second and an appropriate buffer factor based upon the cation concentration. And once the sample has been reached the desired COT value, the end is broken off of the tube of the sample and the solution is blowed into a 100-fold excess of about 0.03 sodium phosphate buffer. And the diluted sample is quickly loaded onto a hydroxyapatite column equilibrated with 0.03 molar sodium phosphate buffer, and at this buffer concentration all the DNA binds to the hydroxyapatite column. Once all the solution has entered the hydroxyapatite column, a 0.12 molar sodium phosphate buffer is added, causing the single-stranded DNA to elute, and the eluent containing the single-stranded DNA is collected in a graduated polypropylene tube. After the single-stranded DNA is collected, 0.50 sodium phosphate buffer is added to the column to elute the double-stranded DNA. Now, the volumes of the single-stranded DNA eluent and the double-stranded DNA eluent are determined, and exactly 0.9 ml of the centrifuge single-stranded DNA is mixed with 0.1 ml of the aqueous 10 normal KOH to denature any DNA duplexes. Now, the sample of the double-stranded DNA eluent is likewise denatured, and the absorbance of both the samples are being determined at 260 nanometer, and the COT value for the percentage of single-stranded DNA has been calculated using the formula VSS into ASS, where VSS is the total volume of single-strand concentration into its absorbance into 100 divided by VSS is the volume of the single-stranded fraction into its absorbance plus VDS is the total volume of the double-stranded fraction into its absorbance, will give you the percentage of single-stranded DNA. And based upon this, you will get a COT curve. So the percentage of single-stranded DNA is plotted on the x-axis, and the log COT value is quoted on the log COT is on the x-axis, and the percentage single-stranded DNA is being found on the y-axis. And based upon this, what happens, you will have a highly repetitive DNA. If you have a highly repetitive DNA, you can see that it is having a small log COT value, and the ones which are having unique sequences, they will have a very log COT value. So if a percentage of single-stranded DNA is being found to be higher, it indicates that it contains a highly repetitive DNA, and it has it reassociates at a very faster rate. As we told, the log COT value is there. I hope this is clear to you. Thank you.